Let's do the TMS. So again, NotebookLM, we have this, they gave it the simulation, the JavaScript, the text around it. We found another 10 relevant sources that loaded all of them. We'll just listen to it real quick, see if we have any comments to make. We could actually try the NotebookLM interactive mode where we could interrupt the podcasting, the broadcasting, ask a question. Let's try that.
Have you ever maybe imagined being able to, well, tune your brain, adjust its settings for better health, maybe sharper focus, or just overall improved performance? It might sound like something straight out of science fiction, right? But today we're diving deep into a really fascinating neurophysiological technique that's actually making incredible strides in, well, pretty much that exact direction.
Yeah.
It's called Transcranial Magnetic Stimulation, or TMS.
That's right.
So for this deep dive, we've pulled together the most important nuggets from a whole stack of sources, articles, some really cutting-edge research papers, even some diagrams to really help us get to grips with this approach. We're going to unpack what PMS is, how it actually works, fundamentally its growing therapeutic uses, and, this is interesting, some truly surprising new insights from recent research that are, well, challenging some long-held ideas.
Yeah, and our mission today really is to help you understand not just what TMS is, but why it matters so much. It has this dual role, which is critical.
Okay.
Both in advancing, fundamental basic science, letting us map and understand the brain's complexities, and in its clinical applications for a whole range of difficult disorders.
Right.
And we'll also touch on the ongoing questions, the debates in this field, because it is evolving so rapidly. It encourages a bit of critical thinking about how we interpret its effects, which are powerful, but also quite nuanced.
Okay, so let's unpack this right from the start then. What exactly is transcranial magnetic stimulation? It's interesting that our sources call it a non-invasive neurophysiological technique, first introduced actually by Barker and his team back in 1985.
Yeah, which feels pretty recent, doesn't it, for something this impactful?
It really does.
But the core principle, the physics, actually goes way, way back to electromagnetic induction, discovered by Michael Faraday in, 1838?
Wow, okay, so how does that ancient principle apply here?
Well, basically, a very brief but powerful pulse of electrical current passes through a coil placed on the head. This current generates a magnetic field, and that magnetic field, this is the clever part, passes painlessly through your scalp and skull to reach the brain tissue underneath.
Right, no surgery, nothing invasive.
Exactly. And once that magnetic field reaches the brain, it induces a secondary electrical field, and that's what influences the activity of the neurons, the brain cells. It subtly modulates them.
It sounds like there's some serious power involved, though. I mean, the stimulator itself, our sources mentioned 3 key parts. These huge storage capacitors, they can discharge, 5,000 amps or more? That sounds like a massive amount of energy.
It is, yeah. A very rapid discharge. Then you have the stimulating coil itself, which shapes the magnetic field, and a very precise timing mechanism to control exactly when that pulse happens.
Okay.
And the design of those capacitors can even shape the pulse itself. You hear about monophasic versus biphasic TMS waveforms.
Right. Does that make a difference?
Well, some research, particularly on repetitive TMS, that's when you apply multiple pulses, suggests that those biphasic pulses might be more important for inducing the longer-lasting changes in brain activity we often aim for in therapy.
Okay, that makes sense. One of the things that really jumped out at early research was how they used single pulses of TMS to study things like motor thresholds.
The MEPs?
Right, using EMG electromyography to actually measure the muscle response, the motor evoked potentials, or MEPs. But what's truly wild is the phosphene threshold stuff.
Oh, yeah.
Where they could actually make someone see a flash of light, a phosphene, just by stimulating the visual part of the brain, the occipital lobe.
Exactly. You're literally creating a visual percept just by applying magnetism externally. It really highlights how directly TMS can interact with specific brain functions.
So how does it do that at the neuron level? What's the immediate effect?
What happens physiologically is quite interesting. A high-intensity pulse causes what's described as a synchronized high-frequency burst of discharge in a group of neurons.
Like they all fire together really fast.
Precisely, a rapid, coordinated firing. But then that burst is followed by something else, a long-lasting GABAergic inhibition.
GABAergics, that's quieting them down.
Exactly. GABA is the brain's main inhibitory neurotransmitter. So you get this burst of activity, then a period of quietening, and this combination effectively disrupts the normal processing in that specific cortical area. That's what leads to this concept of a virtual lesion.
A temporary reversible disruption, like switching that part of the brain off for a moment?
Sort of, yeah. But here's where it gets really interesting, because it's not always just a clean-off switch or lesion.
Oh, okay.
TMS can also just add, well, noisy input into the brain's ongoing activity, like static on a radio line.
Right.
And the balance between creating that clear virtual lesion effect versus just adding seems to depend quite a bit on the intensity of the stimulation you use.
So lower intensity might be more like noise, higher intensity more like a lesion.
That's the general idea, though it's complex. And one study, particularly looking at the visual system, even suggested TMS might be better described as causing a loss of signal, almost like a brief information blackout, rather than just adding random noise.
Huh. That definitely challenges the simple idea then.
It does. It shows we're still refining our understanding. And another crucial point is that TMS is not perfectly focal. It's not like a laser pointer.
Right. Even with the best coils.
Even with a figure-of-8 coil, which is designed for better precision, the effective area you're stimulating is still several square centimeters, not a tiny point.
Okay.
And critically, the magnetic field strength falls off very rapidly with distance from the coil. Meaning the superficial parts of the cortex, the outer layers right under the skull, are much, much easier to stimulate effectively.
Ah, I see. So trying to reach deeper brain structures is harder.
Much harder. And if you crank up the intensity to try and reach deep, you often end up activating the overlying, more superficial areas even more strongly, which can complicate things when you're trying to figure out exactly what brain area is responsible for the effect you're seeing.
That makes sense. There was another fascinating detail in the sources about what part of the neuron gets excited. It's not the main cell body.
That's right. It seems TMS preferentially excites the axons, the long output wires of the neurons.
Okay.
And specifically, it seems to be most effective at points where these axons sort of bend or turn within the electric field induced by the magnet.
Why there?
Because that's where the change in electrical potential along the length of the axon is steepest, what physicists call the spatial derivative. It's like that's the point most sensitive to the electrical nudge from the TMS pulse.
Wow, that's incredibly specific.
It is, and it leads directly into the importance of coil orientation. Just changing the direction you hold the coil, even over the same spot on the head, can significantly change which specific groups of neurons get activated, because you're changing the direction of that induced electric field relative to how those axons are oriented in the brain.
So same spot, different angle, potentially different effect.
Exactly. We see this clearly in studies of the motor cortex changing the current direction, changes the muscle response. And in the visual cortex, it changes how easily you elicit those phosphenes, those flashes of light.
Okay, so location matters, angle matters. What else influences the effect?
A huge factor is something called state dependency.
State dependency, meaning the state of the brain at that moment.
Precisely. The effect of a TMS pulse is massively influenced by how excitable the targeted bit of cortex is at the exact moment the pulse arrives.
Give me an example.
The classic one is in the motor system. If you ask someone to make just a tiny, slight voluntary contraction of the muscle you're targeting,
Yeah.
the motor response, the MEP you measure from the TMS pulse, can be considerably larger, much enhanced, compared to when the muscle is completely relaxed.
So the brain being ready to move makes it more responsive to the TMS.
That's the idea. It really highlights that the brain isn't some static circuit board we're probing. Its ongoing activity, its momentary state is crucial.
But excitability isn't the same as just being active, right? I think one source made that distinction.
That's a really key nuance, yes. Excitability doesn't necessarily mean the neuron is firing rapidly. In fact, neurons are often most excitable when their membrane potential is hovering just below the threshold for firing, but not actually firing yet.
Ah, okay. So they're primed but not busy.
Kind of. If they're already firing at very high rates, their excitability to an external input like TMS might actually decrease. So this whole state dependency thing, it makes interpreting the effects complex. Often it's a bit speculative, trying to pin down exactly why the state mattered in a particular experiment.
It sounds like it. And you mentioned earlier, the effects aren't always localized. They can spread.
Absolutely. The effects don't just stay under the coil. They can propagate through the brain's networks in a couple of main ways.
How so?
Well, one way is pretty direct. The TMS pulse excites neurons. Those neurons send signals down their axons to other brain areas they connect to, causing activity there. Simple signal transmission.
Okay, like a domino effect along the wires.
Exactly. But there's also a more indirect way. TMS can alter the interactions within widespread networks of neurons, even if those neurons aren't directly connected to the stimulated site.
Like changing the background hum or the communication patterns?
Precisely. Think about interhemispheric inhibition, how activity in one side of the brain can suppress activity in the other side. Via connections through the corpus callosum, TMS over one hemisphere can change that balance. So it's not just a spotlight, it really is more like dropping a pebble in a pond and watching the ripples spread across the entire network.
Okay, that makes the brain sound even more interconnected and dynamic. So, given all this complexity, the need for precision, but the limits of focality, the state dependency, what about the tools themselves?
Right, the tools of the trade. The coil design is absolutely crucial because it shapes that magnetic field and therefore the electric field induced in the brain. There are a few main types we use.
What are they?
Well, the simplest are circular coils, but as the name suggests, they tend to induce a non-focal ring-shaped electric field maximum. So the stimulation is quite spread out, not very precise.
Less targeted.
Much less targeted, which is why figure-of-8 coils, sometimes called butterfly coils, are far more common now, especially in research and clinical settings.
Why is that?
Because they consist of two smaller circular loops placed side by side, often overlapping slightly. The magnetic fields they generate add up. They summate right at the junction point between the two loops.
Creating a hotspot.
Exactly. This allows for much more focal stimulation at a limited and clearly definable location. Better precision.
Okay. Are there others?
Yes. We also have double cone coils. These are typically two larger circular windings, but they're angled towards each other, forming a sort of cone shape pointing downwards.
And what are they for?
They induce an electric field that's more deeply penetrating, but also less focal. So you trade some precision for depth. They're used for stimulating areas that are harder to reach with a standard figure of 8, like maybe motor areas for the leg or deeper structures like parts of the cingulate cortex.
Deeper but broader.
Right, and then there are the Hest coils or H-coils. These have quite complex winding patterns and are generally larger. They were proposed specifically for effective stimulation of deep brain structures.
Did they work? Are they better for depth?
Well, their comparative advantage in depth has actually been debated quite a bit in the literature. They are generally less focal than figure-of-8 coils, so you lose precision again. And importantly, they've been associated with a potentially increased risk of seizures compared to other coil types.
Okay, so it sounds like there's always a trade-off here.
There is. This brings us to a really fundamental principle, something often called the electric field depth-focality trade-off.
Okay, what does that mean in simple terms?
It means that for any coil design, physics dictates that if you want to stimulate deeper into the brain, if you want a greater half-value depth, as it's technically measured, you inherently have to sacrifice some precision. The stimulation becomes less focused. The half-value spread increases.
So you just can't have both. You can't have super deep and super focused stimulation at the same time.
Fundamentally, no. It's like trying to get a really tight spotlight beam to penetrate really far. The beam inevitably spreads out. It's a basic physical limitation.
That's really important to understand.
It is, and research backs this up. Figure-of-8 coils are consistently shown to be more focal than circular coils when stimulating at roughly similar depths.
Right.
Now, people have tried to tweak coils, maybe adding things like conductive shielding plates or using ferromagnetic cores to try and shape the field differently.
Does that help overcome the trade-off?
It can alter the depth and focality characteristics a bit, yes, but these modifications don't fundamentally break that basic trade-off limit. Oh, and interestingly, very small coils can be extremely focal. But they would require ridiculously high electrical currents to generate a strong enough magnetic field to actually stimulate neurons, making them impractical for use on humans.
Okay, so we're stuck with this trade-off, but technology must be helping us work within those limits more effectively.
Absolutely. A really significant advancement has been the development of precision targeting and neuronavigation visualization systems.
Neuronavigation, like GPS for the brain.
Pretty much. These systems often use an MRI scan of the individual's brain, co-register it with the person's head in real time using cameras, and show the operator exactly where the coil is positioned and where the magnetic field is likely to be strongest.
Wow.
And now, some cutting-edge systems use sophisticated deep learning models. These models can actually predict the electric field distribution throughout your specific brain anatomy in real time. We're talking fractions of a second, like 0.2 seconds.
That's incredible. So instead of just a generic map, it's personalized.
Exactly. It's a huge leap forward from the older, more static visualizations. It gives clinicians much better, faster feedback to really try and optimize that coil placement for the individual patient. It helps refine the precision we can achieve within those physical limits.
That sounds like a game changer for applying this clinically.
It really is. Which brings us nicely to the next big question. Given all this understanding of how TMF works and these improving tools, what are we actually doing with it therapeutically?
Right. Where is TMS making a difference in treating people?
Well, the therapeutic applications are, as our sources note, rapidly increasing in study. It's become widely accepted as a non-invasive technique.
And where are the biggest successes so far? The sources highlighted a couple of areas.
Yeah, the most consistently favorable treatment outcomes reported have been in depression and Parkinson's disease.
Okay, let's talk about depression first. What are we seeing there?
For major depressive disorder, particularly cases that haven't responded well to medication, what we call medication-resistant depression, lasting beneficial effects have been observed in, well, estimates vary, but around 40% of patients is a figure often cited, which is significant for a difficult-to-treat group.
How does it work for depression? What's the approach?
There are two main established protocols. One involves high-frequency repetitive TMS, usually applied to the left dorsolateral prefrontal cortex, or DLP-FC.
Okay, high frequency on the left side.
Right. The other involves low-frequency stimulation, typically applied to the right DLP-FC. Both approaches have been shown to improve depression symptoms.
Interesting. Why the difference? Left versus right, high versus low frequency?
That's a great question, and the exact mechanisms are still being worked out. But one intriguing line of research suggests that a patient's underlying brain metabolism might play a role.
Metabolism, like how active that brain area is.
Sort of, yeah. Whether the prefrontal cortex shows signs of hypermetabolism being overactive or hypometabolism being underactive, on scans like P scans that might actually predict which frequency approach, high or low, is more likely to be effective for that specific individual.
Wow, so potentially personalized treatment based on brain activity patterns.
That's the hope, moving towards that kind of precision.
Okay, and what about Parkinson's disease?
For Parkinson's, studies have shown some clear benefits. For example, TMS applied over the motor cortex can lead to improved function in the hand on the opposite side of the body, the contralateral hand.
Better movement control.
Yes, and perhaps even more remarkably, applying repetitive TMS over the prefrontal cortex in Parkinson's patients has been shown to actually increase dopamine levels in the caudate nucleus.
Dopamine, that's the key neurotransmitter lacking in Parkinson's, right?
Exactly. So TMS seems to be able to modulate that crucial dopamine system. Now, the sources often describe the overall benefits in Parkinson's as perhaps modest.
Yeah.
But for patients managing a progressive condition like PD, even modest improvements in motor function or potentially boosting dopamine can be really significant for their quality of life.
Definitely. Are there other conditions where TMS is being explored?
Oh yes, quite a few. It's being actively studied or applied for a whole range of other neuropsychiatric conditions.
Like what?
Things like task-specific dystonia, like writer's cramp where a specific movement becomes impaired. Also, tech disorders like Tourette's, certain types of muscle jerks called cortical myoclonus, even conditions like intractable seizures, schizophrenia, chronic neurogenic pain.
Wow, that's a broad list.
It is. Also spasticity after stroke or injury, and using TMS to try and influence recovery after stroke.
So, lots of potential. But are the results as strong as in depression and Parkinson's?
Generally not yet, or the effects might be more variable or short-lived. While there are definitely positive results reported in many of these areas, the effects are often described as transient. They don't always last very long after the treatment stops.
Okay.
And for many of these conditions, the consensus is that we still need more well-controlled, multi-center, randomized clinical trials, basically larger, more rigorous studies to really nail down which conditions TMS is definitively effective for and what the best treatment protocols are.
Right. More evidence needed for a wider application. But going back to depression and maybe PTSD, you mentioned earlier how TMS can affect brain networks. That sounds really key.
It is. I think this network perspective is where a lot of the really exciting current research is happening. We're moving beyond just thinking about stimulating one spot and looking at how TMS influences the communication between different brain regions.
So it's like retuning the whole brain's communication system, not just one part.
That's a great way to put it. The idea is that in conditions like major depressive disorder, MDD, or post-traumatic stress disorder, PTSD, there are often patterns of pathological neural network functional connectivity. Certain brain networks might be talking too much to each other or not enough.
And TMS can adjust that.
That's the theory, and the evidence is growing. TMS therapy seems able to modulate this abnormal network connectivity.
What kind of specific network changes are they seeing?
Well, some fascinating findings are emerging about predicting who will respond well to TMS based on their pre-treatment network patterns.
Okay.
For instance, one study found that having more negative connectivity, meaning the activity in these areas, tends to go in opposite directions between a region called the subgeneral anterior cingulate cortex, or SGACC, often implicated in mood, and the default mode network, or DMM, a network active during rest and self-thought. Having more negative connectivity before treatment actually predicted better clinical improvement after TMS.
So a specific brain signatures predicts success.
It seems so. And conversely, another finding was that having more positive connectivity moving together between the amygdala, key for emotions like fear, and the ventromedial prefrontal cortex, involved in regulating emotion, that also predicted better outcomes.
Interesting. So it's about the specific links, not just overall activity.
Exactly. It suggests that maybe having a brain network configuration that's already in some ways closer to a healthy pattern, perhaps a less overconnected DMN or stronger prefrontal control over the amygdala, might make someone more receptive to the therapeutic effects of TMS.
And what about after TMS? What network changes correlate with getting better?
Yeah, looking at the mechanisms of action, studies have found that symptom reduction, feeling better, was associated with reduced connectivity between that SGACC region and several key areas, including the DMN, the DLPFC, the spot often targeted, and also the insula.
The insula. What does that do?
The insula is involved in lots of things, including interoception, sensing your internal body state, and integrating emotional and cognitive information. It keeps popping up in these network studies.
Okay.
And another finding was reduced connectivity between the hippocampus, which is vital for memory, and the salience network, or SN, which helps detect important external and internal stimuli.
So it seems like TMS is dialing down some potentially problematic overconnections.
That appears to be a key part of it, yes. Particularly dialing down the influence of the SGACC on other networks and maybe modulating how emotional memories interact with attention systems. And that consistent finding about the insula, it's making researchers think that maybe directly stimulating the insula itself could be a promising avenue for future therapies.
That's fascinating stuff, seeing the brain respond at that network level. But this brings us to something you mentioned earlier, this idea of TMS being purely excitatory or inhibitory. That's being questioned now.
Yes, and this is a really important point, almost like debunking a long-held myth according to some of the newer perspectives we looked at.
Okay. What was the old idea?
The early work, especially with repetitive TMS, seemed to suggest a simple rule. High-frequency RTMS was thought to be facilitatory or excitatory, increasing brain excitability, while low-frequency RTMS was thought to be inhibitory, decreasing excitability.
Right. That sounds like a neat, simple picture.
It does, and it was a very influential idea for a long time. But contemporary studies, looking much more closely at the complex effects and considering state dependency, they really challenged this stubbornly persistent assumption, as one paper put it.
So it's not that simple. High frequency doesn't always excite, low doesn't always inhibit.
Exactly. The current thinking, or at least a strong emerging view, is that no RTMS protocol is by its nature either excitatory or inhibitory.
Okay, so what is it doing then, if not just turning things up or down?
Its effects can be caused by a whole range of mechanisms other than just simple binary changes in overall excitability. It might be changing the timing of neuronal firing, altering synaptic plasticity in more complex ways, affecting different types of neurons differently, influencing those MEPOC oscillations we talked about.
Right. Much more nuanced than just a volume knob.
Much more. So researchers are now being urged to use real caution with those simple excitatory and inhibitory labels. They might be misleading because the reality depends so much on the specific protocol, the brain region, the brain's state at the time, all those factors we've discussed.
So if we can't just label protocols as excitatory or inhibitory, does that make it harder to design treatments? Or maybe, does it actually open up new ways of thinking about how TMS achieves its effects?
I think it's more the latter. It forces us to think more deeply about the mechanisms. It emphasizes this complexity and the ongoing evolution in our understanding.
But it doesn't mean TMS isn't effective.
Not at all. Despite these nuances, TMS absolutely remains a powerful and valuable non-invasive tool.
Okay.
So the overall conclusion, I think, holds firm. TMS is an effective tool, both for fundamental research mapping, brain function, understanding connections, and for a growing list of therapeutic uses.
Yeah.
Its study is booming. The outcomes for depression and Parkinson's are particularly encouraging, and there's continuous promising research for many other conditions.
But we still need more research.
Definitely. We still need more insights into the precise physiological basis of its effects, especially the therapeutic ones. We need to better understand that causal link between the TMS intervention and the clinical improvement. And yes, as mentioned, those well-controlled, multi-center randomized clinical trials are still crucial to definitively establish its role for many conditions, but the trajectory is undeniably positive.
What an incredible journey we've taken today. Really getting under the hood of transcranial magnetic stimulation, from the basic physics, Faraday's induction,
Right, from 1838.
to how it interacts with individual nerve cells, those axons bending in the field, the state dependency, the network effects, and then these cutting-edge therapeutic applications constantly being refined. It really feels like a technology that's reshaping possibilities in neuroscience and mental health care.
It truly is. And maybe a final thought to leave our listeners with. Considering that fundamental depth-focality trade-off we talked about, you can't easily get deep and focused.
Right.
And considering the really complex, state-dependent ways TMS interacts with brain activity and whole networks, how might future research leverage these increasingly sophisticated tools like personalized neuronavigation and real-time e-field modeling?
Yeah, how can we use that?
Could we use them to truly optimize individual treatments, moving beyond those simplistic excitatory or inhibitory labels, maybe by tailoring stimulation based on an individual's real-time brain state or network activity, and perhaps even understanding better the role of things like expectation or placebo effects, which also likely engage these brain networks? Could untangling all that unlock even greater potential for healing?
That's a fascinating challenge, really pushing the boundaries of personalized neuromodulation. It's clearly a field brimming with possibilities, and we definitely encourage you, the listener, to keep exploring this rapidly advancing area of neuroscience. It's going to be exciting to watch.
Okay, love the excitement. But we also shared an interactive TMS brain simulation tool. Did you cover that one?
That is such a great point.
Oh, yes.
We did mention the neuronavigation visualization system.
The real-time e-field modeling.
But you're right, we definitely shared an interactive simulation tool from BioniChaos.
And slicer TMS, too.
Yeah, that's exactly what we were thinking about when we talked about personalizing treatments.
Those tools are incredible for visualizing the TMS effects in 3D.
You can actually explore different coil types, like the figure 8 or circular.
And adjust stimulation intensity and frequency, all in real time.
It's like having a little lab right there.
It truly helps illustrate that depth-focality trade-off we just discussed.
Yeah, you can see how the field spreads out.
And how precision targeting allows clinicians to really try and optimize that coil placement.
It's a huge leap from older static images.
It also highlights how rapidly this field is evolving.
And how important those visual aids are.
For both researchers and clinicians.
It's going to be exciting to watch.
Absolutely. The way those tools predict electric field distribution in fractions of a second.
Yeah, it's just mind-blowing.
It really streamlines the planning process.
So those simulation tools are definitely a key part of this future.
It helps bridge the gap between complex science and practical application.
And gives us hope for even more tailored approaches.
For individual patients, based on their unique brain anatomy.
It's all about that personalized neuromodulation.
Exactly.